Thank you. Good morning. Today, I will talk about the origins, functional relevance, and limits of scale-free temporal brain fluctuations. And I start by saying that, as some of you know, I've been working on these subjects first with the theoretical training by Paolo Gregolini, who is here, and then by applying statistical methods to the study of this aspect of brain activity quite a long time ago. And then I delved myself into studies of cognitive neuroscience. Yes. And I took the opportunity of this conference to come back to looking at these aspects of the brain activity, which are typically overlooked in cognitive neuroscience studies. And I say this because there will be more questions than answers at the end of my talk, and these questions are for you. And also because, and I say this also because I will start from one of my studies on cognitive neuroscience, because this gives us a hint on how this part of the brain activity has been overlooked. So what I study is I use electrophysiology, in fact, the EEG, to study the neural basis of perceptual and cognitive predisposition in infants, and in particular in newborns in the first 3 days of life. And here I briefly present a study that we did a few years ago about the cortical response to faces in newborns. So newborns do not have any visual experience. They just, and in the first 3 days, they have really little visual experience. So this study was to test whether there is some kind of predisposition to faces, which has already been demonstrated behaviorally. So just to make the long story short, what we present is the simplest possible representation of a face, which is just the shape of a face with 3 dots, with the upper 2 dots representing the eyes and the lower dot representing the mouth. And then as a control condition, we present a perceptually equivalent stimulus in which the only difference is the fact that the orientation of the dots is inverted, disrupting the face configuration. In cognitive neuroscience, what we try to do, is to design an experimental presentation such that we are able to extract the neural response related to the stimulus as reliably as possible. And in this case, what we did, I can explain, the details, but I will go fast here, is to present these phases with a very specific periodic stimulation. So we present these phases that appear and disappear very slowly, 0.8 hertz. This is to match the rudimental visual system of the newborns. And we do this because we want to drive the stimulus, and we want to know in the noisy EEG and very complicated EEG of the newborns exactly where to find the response. So if we look at the power spectrum, of course, that's we are going to look in frequency of the response. So here we see immediately that at the frequency of stimulation, 0.8 hertz, you see the red circle points to the peak in frequency. So the neuroscientist is very happy with that because we can find the response to our stimulation. But the physicist maybe thinks in another way and says, okay, of course, you drove the stimulus with this frequency, and this is a trivial result. But what you can see is that, so the dashed line that you see in the power spectrum is a power law. So you can see that the power law profile, apart from the peak, is very well fitted by a power law. And so the physicists can say, this is the interesting part, because it's a power law. So it could be scale-free fluctuations, there could be scale invariance, and maybe a sign of self-organization. So this is just to show that even in the standard cognitive neuroscience studies, this power law profile appears. So just briefly mention the result of the study, and then I will go to the subject of the power law. So this, if we compare the peaks to the phase with respect to the inverted phase, we find a very strong activation, which then we reconstruct anatomically, and we find the phase processing network that you also find in adults. But back to the scale-free dynamics. So as I say briefly, everyone here knows that I will show is evidence of power spectrum decaying, going as f to the minus gamma for frequencies tending to 0, and in a couple of studies, I will also mention the trend fluctuation analysis, which is another way to basically look at the same kind of behavior. And here, what I will look at is, first of all, is scale-free dynamics in the brain really homogeneous across all levels of the brain organization, or are there variation in the scaling exponent and also in the frequency range of the scaling exponent? This is important. Let's really observe the data and see what they say. Second important part is what property of the brain really does the power law, the scale-free fluctuations reflect? This is really an open and recent question. And the third question, which I anticipate that I won't find the answer, but I will ask you the answer, is it an epiphenomenon of spatial temporal filtering, or does it reveal some kind of self-organization of the system? And how can we find the answer to this response? So in order to understand what I will show, just a very brief outlook of all the kinds of electrophysiological recordings that we can do in the brain. So as you can see here, I think you can see in the bottom, you see the layers of the cortex, and the most microscopic recording that you can do is to stick an electrode in the neurons, do intracellular recordings, and you can see the single spikes or the membrane current. Okay, grazie. And oops. Okay. Here. And then you can see local field potentials, which are recorded outside the cell. So they record the activity of a certain number of neurons. And then you can also have electrocortical recordings, which are recordings on the surface of the cortex, but inside the skull. This is also another level of spatial integration. So they integrate to more neural populations. And then you have non-invasive electroencephalographic or magnetoencephalographic recordings that record wider areas of the brain. So that's why I say recordings at different spatial integration level. Now, I will give you an overview of the power law of the scale-free brain fluctuations at all these levels, starting from the membrane potential. Here you see the power spectrum of the membrane potential. This is one study showing that the range of power law is relatively large, from 10 to 500 hertz, more or less. And the exponent is very high, 2.6. I will come back to that. And this same behavior is found also in local field potentials and also in electrocorticographic recordings. On the other hand, if we go on the top, on the surface of the scalp, we see with EEG that we still have a power law of the power spectrum, but this is in a lower frequency range and with a lower exponent, significantly lower, which also we can find with the trend fluctuation analysis. And if we compare EEG and MEG, that are similar ways to measure the same thing, we see that the exponents are between 5 and 1.5, more or less. So to assume we have really a difference here. Intracranially, we have an exponent between 2 and 3, which is highly non-stationary, and and a relatively high frequency range. While for EEG and MEG, we have a lower exponent and a quite restricted frequency range. Okay, take this as this information. And now the second part, which is is there any relation between this power law profile of the power spectrum and the neural activity, local or global neural activity? And one finding that has been also reproduced in the years is by people simultaneously measuring the spikes, so the firing rate of the neurons and the local field potentials. So inside firing rate of the neurons and the local field potentials around the neuron. And what they found is that the firing rate is proportional to a broadband shift of the power spectrum. So the power spectrum keeps always the power law profile, but there is a shift. So more higher firing rate, higher shift. And this has also been found at a slightly higher level on the motor cortex, such that this measure has been used to decode the finger representation. So if you look on the motor cortex exactly where a single finger moves, the way to understand whether it moves or not is to look at the shift of the power spectrum, the shift of the power spectrum. Okay, this is one mechanism. The other mechanism we can find already in cortical layers. In this work, they measured the power law profile of the power spectrum in different cortical layers, and I just point to the two extremes. The superficial cortical layers have typically a lot of feedback modulation. Feedback modulation for other parts of the brain are some slow modulations, so low frequency inputs, while the deep layers, the red ones, maybe I point it, so this is superficial layers and this is deep layers, they have the input from the feed-forward input from the sensory areas, which are typically more high frequency. And what we find here is, again, is the modulation of the slope in this case. So all these data have power law profile of the power spectrum. This is maintained, but what changes is the slope. So in this case, it looks like that it depends on the input. If the balance between the low frequency part and the high frequency part of the input changes, then the slope changes, but the power law is maintained. And we have similar effects at the non-invasive level of EEG. So with the exponent rising, for example, with sleep, and this is also consistent with this idea that sleep brings some more low frequencies in the data, and also with anesthesia. And this work shows that there is hypothesizes that in fact this power law signals the balance between excitation and inhibition in the brain. Because by manipulating two different kinds of anesthesia, it shows that when excitation goes up, the exponent goes down because excitation is shorter time scale, while the opposite occurs when you have more inhibition. And the last evidence that I show is a work that I did 15 years ago with a reasoning task. And this is the trend fluctuation analysis, but again, so I'm going to be very brief. The idea is that we have a subject in two conditions. One condition is that he has the feedback that he found a rule in some kind of numbers, so he just has to confirm the rule. And in another condition, is we tell him, no, the rule is another one. You have to find out the good rule. So it comes back in time and has to re-elaborate all the information that he had. And this second condition has a higher exponent than the first one. And again, I think this is dependent on the balance between low and high frequencies. Angela, how many? 7 minutes. Okay, good. Okay, so I gave you an overview, which is pretty complicated, but let's try to get some ideas from here. In order to explain this data, there have been quite a number of researchers who tried to do models of 1/f brain dynamics. And these are biophysical models. So these are models that just try to understand do a model of a source or of electrical activity, like this one, and then try to understand how it spreads through the neural tissue. And a series of models were done by Bedar and Dextex quite many years ago, and they're still refining their models more and more. And I'm not going into the details of the model because they are really complicated, but the result is that by changing the parameters of these models, they can recover a power law function of the power spectrum in selected frequency ranges. So these models are successful in reproducing this kind of pattern. Of course, they don't contain any kind of memory, any kind of self-organization. It's just sort of models starting from Maxwell equations. Another series of models were done to try to understand whether this idea that the balance between excitation and inhibition could modulate the power law, the power spectrum. And as I mentioned before, here the idea is that the inhibitory activation has a slower time scale than the excitatory activation. That's why when the inhibitory wins, the exponent goes up because we have more lower frequencies, while if the excitatory goes up, where we have more higher frequencies. So again, this is important in particular for psychiatric disorders for which we have hypotheses of imbalance of excitation and inhibition, because this could be a very nice proxy. Okay, now I sum up some answers to my questions. First of all, scale-free brain activity is ubiquitous because it's everywhere in all the recordings, but within very restricted frequency ranges, in particular for the EEG and MEG. Second, frequency ranges and exponents decrease substantially from intracranial to scalp level recordings. This is something that has to be explained. And third, scale-free brain activity reflects both local neural activity and balance between low and high frequency activations. These are important results. And there are models that explain these results just by modeling the filtering properties of the neural tissues. Now, the open questions. These are for you just now or in the following days. I will be happy to discuss them with you. First, and some of these questions do not only pertain to brain activity. They pertain to any kind of signal, physiological signal, geological, or of any kind that exhibits power law like power spectrum. Is a 1.5 decade of power law sufficient to be elected scale-free? How important is the extension of the frequency range in which we have scale-free fluctuations? What kind of dynamical system generates a power spectrum scaling exponents with scaling exponents larger than 2? Because between 0 and 1, we are in the ergodic regime. Between 1 and 2, we are in non-ergodic regime, but still it works. But for larger than 2, I don't know what is really the underlying dynamical system. Are filtering models that I showed to you in contradictions with models based on self-organization? If no, I mean, can we reconcile this kind of two models? If not, can we disentangle them and test whether it's the two is one or the other? What is the cause of the decrease of the exponents and frequency ranges from inside to outside the skull? This is more like a specific to the brain, in fact. And the big question is this scale-free dynamics an EP phenomenon, or does it have some functionally relevant properties? So is it generic, or does it really signal to us the brain activity as a specific properties and a specific structure? And the very last question, and then I close, is coming back to the work I showed to you, in which I look at the peak of the power spectrum at the stimulation frequency. I ask myself, after all these modulations, is the background power spectrum really unrelated to the stimulation, or is the whole power spectrum shifting or rotating with any neural processing? Should we reconsider this as the background power spectrum, or should we always, sort of change our approach and look at it as also a part that is modulated by the neural activity. Okay, thank you very much.
Thank you.